assuming that means you can all hear me. All right, so I'm recording now, and just a reminder that anyone who needs to get into lab today is, hopefully we'll be able to fit everyone who needs to get in there today. It's not limited to, a certain group. So between the teaching lab and my research lab and the core, we can spread out if need be. So, and then the other thing I wanted to see if you'd be interested in, so I held a open lab session yesterday afternoon, and a few of you took advantage of that. And then I was thinking maybe one of the evenings this week I could come in for a little bit if that would help anyone. So today's Monday. So we, obviously we have lab, Monday morning and Friday morning. What other day, evening, like late afternoon, early evening, would work for people, do you think? Any suggestions? Thursdays? Anyone else? Anyone via Zoom? Anything there? What day of the week might be handy for you to come in and make progress?
Like Tuesday or Wednesday.
Tuesday or Wednesday?
Thursday's perfect for me.
Thursday is the only time what? Yeah, there's classes, there's other classes in there, you mean? Or like the open lab? Oh, you're talking about the other open lab sessions. Is that what you're talking about? Ah, okay. So we were just saying in person here that, Jay-Z and Alex and Rachel have those open times, but Thursday is the only time that they are not doing anything. So from that perspective, that might make the most sense, because remember, you can go in there during those open times that the med micro teaching instructors are just in there having an open session. So make sure to take advantage of that. I've told them to be aware of you coming in, and they know that. So then maybe Thursday would be another time that I could come in to cover that day. And then of course we have lab on Fridays. So I'll have to look at my schedule. I have a busy week this week. But I'm guessing it would be Thursday, late afternoon, maybe around 5, but let me double check that and then I'll send out an update. But tentatively Thursday, late afternoon would be an option for anyone who wants to come in and do some more work on your lab research project. Okay. So getting back into microbe-animal interactions, I think we left off, we were looking at this idea of the ants and the attine ants and their fungal gardens and how they keep it so clean and pest-free. And we saw that there were a couple extra players in the game that they discovered, besides what the microbes that were on the ant exoskeleton, the beneficial microbes that are able to produce antibiotics to inhibit the fungal pathogen, the parasite here on this image. So that's Escovopsis pathogen. And then they found the black yeast also hanging out. Subsequent studies to this one have now shown that the black yeast isn't always present depending on the ant colony and the ant species, but it has been shown to show up in several instances. Then they've been continuing to study this. They found the nitrogen fixers, so that was an interesting addition to the story as well. I'm not sharing my screen. Well, that would help. Although so far what I've talked about, you've all seen already. But let me Now it won't let me do what I want to do. How about now? Do you see one big? Okay. All right. So the nitrogen fixer is showing up as well. And so, you know, all this research was being done before the human microbiome project really kicked into gear. And so this has, you know, they've continued to study this whole interaction, and they're realizing that there are more players every time they look, in the sense of, you know, truly there is this kind of microbiome idea going on with the ant and the fungal garden, and that there's several different organisms. And then they started looking more carefully at the pathogen, the Ascovopsis pathogen, and realizing that it is no slouch when it comes to being able to interact and defend itself, and trying to get a foothold into these fungal gardens. And so this was just published this year, looking at chemical, chemical warfare. And so you have the beneficial microbes that are doing the hard work of trying to inhibit the pathogen and keep that fungal garden pest free. But then the pathogen is able to both physically attack both the insect, so it has some interesting chemicals that it can produce to weaken the insect, the ant, and also to attack the fungal garden and the bacteria that is trying to inhibit the fungal pathogen. So the Ascovopsis is learning to fight back, and probably has been for quite some time, but this paper did a really nice job of looking at these interactions, both beneficial with the beneficial bacteria, but then how the fungal pathogen is also fighting back. All right, so here, this diagram is trying to get at this kind of the idea of that the fungal pathogen, they now know, can really actively send out chemicals that can inhibit the beneficial bacteria, that can actually directly start impacting the insect, the ant, as well as ways to get into the fungal garden and start to gain a foothold. And so there's this, the red means that there's inhibitory interactions going on of some sort. The green means there's beneficial. And then they started, in this paper, they were looking at what types of chemicals might be produced. And so with this one, they're looking at just this image is just focusing on what is the fungal pathogen doing, what is Ascovopsis doing. So you see all these chemicals, and you can see, you know, the big picture idea is that there is an array of chemicals that the fungal pathogen is capable of producing, and they're trying to show just by the different colored images here with the different shapes that there's different chemicals being that will target different players in the game so that the fungal pathogen can try to get a foothold. And so again, you see, you know, there's the pseudonocardia, the streptomyces, so those are the beneficial bacteria that hang out on the insect, on the ant. And so you have all this interaction going on, this image just trying to show what the fungal pathogen is up to. And then here you're looking at the idea of, you know, how all the other organisms are trying to fight back against the pathogen, but they're also realizing that some of the beneficial bacteria may be able to also kind of have their own little battle going on to some extent. And so they're exploring this idea too of, you know, are the beneficial microbes working together, or are they also trying to like get, you know, the upper hand on each other to gain more space and nutrients? So that this is a complicated story. The more they look into it, the more complicated it seems to get, which of course that's what science usually does, right? And so here you see a big mess of different chemicals. These are from the beneficial bacteria now. So they're really getting into the chemistry of this and exploring the different types of antibiotics that are being produced. And as you can see, wow, you know, pretty complex molecules. This is one reason why we tend to still use streptomyces and related bacteria to produce our own antibiotics that we, you know, take as humans and animals, because you look at some of these antibiotics, they're very hard to make synthetically. The pathway to get those made, we still rely on the bacteria to do that rather than trying to figure out how to do it from scratch in the lab. So the story continues. This is a 2020 publication, and again, just getting at this idea that the interactions are more complicated than what they originally thought, and they're realizing there's many players in the game and many interactions, some very positive and some definitely aggressively negative. And they're trying to tease apart what's going on with the beneficials. I find that interesting, what's going to happen with like the streptomyces and pseudonocardia. How much do they fight and how much might they cooperate? And so I don't know, I have not found any papers on quorum sensing. I was interested to see if maybe the beneficials could be doing any kind of quorum sensing, but I don't think anyone's looked at that yet, unfortunately, because I would think if they're living that closely to each other on the insect exoskeleton, if they're physically near each other and they're coexisting, chances are fairly high that they're doing some kind of quorum sensing and cooperating with each other, but no one's looked at that yet. So that story is still to be told. Okay, so microbes, some of the early studies when they looked at different groups of insects, they did find that there are microbes there either living on the insect or a lot of them focused on what's going on in the gut or the mouth part of the insect. And especially when you look at this interaction, the bacteria and aphids. Aphids are they're very detrimental plant pathogens. They're able to attack all kinds of different plants, and they don't really, the damage that they do is that they are able to very precisely pierce the vascular tissue of the plant and suck away the sugar water. So as the plant is busy making sugars, the aphids can pierce right into the vascular tissue and start taking that away. So of course that starts to weaken the plant. Well, then sometimes they also introduce different microbes as they're feeding the mouth stylet proboscis part. Can actually kind of allow for bacteria and other pathogens to get into the plant. So it's almost like it's injecting it right into the vascular system. So it's a very precise way to introduce microbes. And then because it's a vascular system of the plant, the microbes could go throughout the whole plant body and potentially survive and set up camp and start causing disease on the plant or in the plants. And so this, of course, the aphids in and of themselves are causing problems because they're weakening the plant by taking away the sugars, but then they're also capable of being a very precise way to introduce plant pathogens that can cause problems. So aphids are tough to spot. If any of you have been in our greenhouse and have been working with plants in there on any research projects, sometimes aphids do show up in our greenhouse, but you have to really look closely. They tend to hang out on the underside of leaves. And so you would have to turn the leaf over and look very closely. Often they're the same color as the leaf or just slightly lighter in color. Here they're looking like a yellow-green, and they're very, very small. This is a blown up picture. So you have to be looking very carefully to spot them. Another issue with aphids is that they mutate quite rapidly. And so all of the, well, I shouldn't say all, I don't know that for sure, but so many of the pesticides that they use on greenhouse aphids, especially greenhouse aphids, we see that they mutate much quicker because they're getting constantly exposed to all these pesticides and they gain resistance to it, and then they multiply, and produce babies, and they're all resistant to it. So then you hit them with a new type of pesticide and the cycle keeps continuing. And so the greenhouse aphids in particular, compared to wild native aphids that are just out on in living their life outside, there's a difference clearly in their resistance patterns to different pesticides. And so they're a big problem. And there's many species of aphids. And so the phloem vessels are the vascular part of the vascular tissue. And that the phloem is where you see the sugar coursing through the plant body. And if you dissect aphids, you'll see that they have this mycetome tissue in their head region and it is a house essentially for bacteria to hang out inside. So Buchnera is one that they've identified. Now this one is interesting because Buchnera, if you give the aphid an antibiotic that kills the Buchnera bacteria, the aphid will die. So this is a very much almost an obligate relationship in the sense that the aphid needs Buchnera. And so when they looked at, okay, why is it so dependent on Buchnera? And when they did that, the bacteria are producing certain amino acids, AA amino acids, that and other growth factors that the plant, the aphid's not getting from the plant. The plant is basically, the aphid's basically getting a lot of sugars from the plant and maybe a few other things, but it's not a complete meal. It doesn't have a full repertoire of everything it needs to survive. So the Buchnera bacteria is providing those extra needed nutrients for the aphid to survive. So in this case, we see kind of a complicated thing. This aphid is it's a pest, but it can also introduce pathogens into the plant. And then we see it also has its own suite of at least this Buchnera that hangs out with the aphid to keep the aphid alive. And so there's, again, a complicated story when it comes to the aphid plant pest. So there's a close-up. Kind of cute in some regards, but sure don't like to see them in the greenhouse, that's for sure. And then here is under the microscope, they're looking at mycetome tissue. And so if these two they almost look like little glands here, that's the mycetome tissue. And then the S is salivary glands. The P is the proboscis that comes out of the head region of so this is a leech instead. I couldn't find a picture of an aphid. But they are finding that pathogens that suck sugar water out of plants have this, and then the blood-sucking insects like leeches, if you've ever had a leech on you, or lice, I suppose, there's bacteria hanging out in there as well. Did you have a question, JZ?
Yeah, so Mike doesn't talk about how they use aphids to what's in the farm. If they try to use aphid to inject any specific microbes into.
Oh, yeah. So the question was, aphids are so precise at getting their mouth part, their proboscis, to precisely insert into the phloem tissue. And so could we use them as a delivery vehicle, right? To deliver, like I've seen people actually inject plants with hypodermic needles with antibiotics. Like they'll do that to introduce an antibiotic. So it'd be fun to like genetically engineer aphids to bring good things to the plant, maybe some biocontrol agents or some kind of chemical that would help the plant survive a current crisis it's in. Yeah, I don't know if anyone's looked at that, but they would be a cool vector, because they're so precise. And so that would be one way to easily inject something into the phloem tissue. You just got to stop them from sucking away all the sugar water. This is just under the microscope when you smash some mycetome tissue and look at it under the microscope, and you can see these squiggly long rods. That's Buchnera species. I don't know if it's Aphidicola or not, but it's some Buchnera species. So plenty there hanging out. And then we kind of talked about this already with this idea of you can have eukaryotic organisms. They're not really animals per se, I guess, but eukaryotic organisms that have their own bacteria inside. So they're often single-celled eukaryotic. Protozoa are eukaryotic. Typically, they are single-celled or exist in small colonies. And you can see a very nice image here showing all the orangish, that's all the bacteria hanging out inside. And so there's scientists that are studying this carefully because they're easy to work with, and this is such an intimate association. So they're studying this dynamic to just get a little bit better idea of what might be happening in the process of evolution And especially, there's been a lot of questions about the organelles that we see inside eukaryotic organisms. I mean, we already seem to have enough data to support the endosymbiosis theory with how our, the cells that we have, when you look at the mitochondria, they were once thought to be free living bacteria, ancient bacteria, same with chloroplasts in plants. Well, they've also been looking at the nucleus, they've been looking at flagella, all in other internal organelles that they were once, perhaps, could they have been free living microbes that came together into what we now call the eukaryotic cell type. And so there's still a lot of work going on in this area. I don't keep up with it real steadily, but when you go to microbiology conferences, there's usually some symposia sessions on, this idea. So the endosymbiosis idea is kind of not finished. They're still looking at these ideas. Medical applications, of course, if we could understand some of this better, how could we use that to help with human disease? And so these interactions, could we design beneficial microbes that humans could take that could persist in our body long term? And now with the Human Microbiome Project, that idea is gaining a strong following, and actually there's businesses popping up looking at this idea. One of my former grad students was working, I think he's still there. He was in Minneapolis area working for a company that was designing probiotics they initially started their company for C. difficile infections, ways to prevent that. And they've branched out into other diseases as well. But again, so this Human Microbiome Project, what we're looking at here is acanthamoeba is the big green protozoa, and then the protochlamydia bacteria. That's what the image you're looking at there. So this intimate interaction here between bacteria living endosymbiotically, they're going to, that's something, again, the story is not finished. And so the work that Dr. Lynn Margulis started here back in the 60s and 70s, they're still working on it today with some of these other organelles and parts of the eukaryotic cell. So there is this, the more they started looking, the more they realized, there's all kinds of examples of microbes hanging out in other animals. So if you go to the coral reef system, it is just prolific examples of microbes that are hanging out on and in these different organisms that make up the coral reef. And it can be bacteria, it can be algae. It just depends on the organism you're looking at. But again, it's such an intimate association that it's definitely a win-win. When you think about it, the endosymbiotic organisms, whether it be algae or cyanobacteria, they're gaining a pretty safe home that's pretty steady in terms of its, you're in an enclosed organism, so you're safe and you don't have huge swings in your environment. And then if you're the coral or the hydra or the anemone, of course, if you can have a being inside of you that can do photosynthesis and give you all those sugars, that's pretty great. All you have to do is point yourself to the sun and you have your partners doing that lovely job of photosynthesis for you. So there has been a lot of really interesting studies on coral reef systems. There is, I don't think it's in this slide set, but it's in another one that we'll be looking at soon. There's a whole society that popped up when they started really getting into this idea of kind of the microbiome of different organisms, and it's called the International Symbiosis Society, which I used to belong to. I forgot to up my subscription this year, but anyways, they have conferences every 3 years where and a lot of what they focus on is on the coral reef systems. That's one, because there's so many different species there that are, have their own little microbiome. And of course, now with the coral reefs dying off, they're trying to figure out what's going on with that and the impact that has not only on the animals there, but then their associated microbiome. So they've looked at corals and dinoflagellates. Dinoflagellates are kind of like a special type of algae. And so they are looking at, what are the partners doing for each other? Of course, if you're an algae, you can do photosynthesis, but you can also take up certain chemicals that are considered waste, like ammonia, and convert that. And then, of course, if you're the animal, the coral, you can provide protection, and they actually can extend their body, their polyps, toward the light. And that helps, of course, the algae do photosynthesis. And so they're a really interesting dynamic. And they're showing that clearly the effort of the algae in the coral is very important, because the coral growth is directly correlated, positively correlated with the activity, the photosynthetic activity of the algae inside the coral. And so again, thinking about this idea of the coral reef organisms and their microbiome, they're trying to, figure out as much as they can about the health of the coral reefs. And now they realize they really do need to look at the microbiome and factor that in as they're trying to race against the loss of the coral reefs dying off. Some really pretty pictures of, you can see the polyps being extended. You can see the top image, really nice example of the algae there cooking along, doing photosynthesis. You have some brown algae here as well, I believe, that are helping. And so just anyone who's been fortunate enough to go snorkeling or scuba diving on a coral reef, I have yet to do that. I've only scuba dived in Lake Superior, but if you ever get the chance, pretty fascinating. Fungal? Wasn't that, at one point, wasn't that a hypothesis for part of the reason the coral reefs were dying off, is that there were fungal pathogens in a negative way? I don't know about a beneficial. Yeah. I don't know. I have not seen any reports of any fungi, beneficial fungi. I have not come across that yet, but I wouldn't be surprised if somebody's trying to look for that and trying to see. Because again, we are realizing that the health of the animal is highly dependent on the microbiome. And so if something's happening to the beneficial microbes, and that could be part of the reason why the coral reefs are dying off, is that the microbiome is getting hit pretty hard. So again, lots to understand, and it's kind of a race against time here, trying to deal with the coral reef system and declining health. And then if you want to go even deeper beyond the coral reef and go deep into the ocean, you can witness these creatures, which are looking at the idea of these tube worms. And so the whitish and then the orange top, those are the tube worms. Here you see a fish swimming around too. So this is a hydrothermal vent. And if you've ever seen videography of the submarines that can get down that deep and they are showing footage, you'll see like this, they'll be, chugging along in their little submarine, and it'll just be kind of this moonscape for a while. And then all of a sudden they'll come upon a hydrothermal vent. And all of a sudden there's this huge increase in organisms that are hanging out in those areas. So it's a very patchy distribution of who's living down there and where they show up. The hydrothermal vents tends to have very high populations of organisms hanging out in that area. And that's probably because it's so rich, as the hot water coming up through the crust of the earth there and coming up into the water of the ocean, it brings in all these minerals into that area. And as we know, there are plenty of microbes that can handle really hot conditions down there. And so now we're thinking about these primary producers, the chemolithotrophs, so that yes, they're not, they're not going to be doing photosynthesis down there because it's too dark, but they can still be primary producers where they can take inorganic material and CO2 and, end up with sugars and ATP. And so if you're an animal living down in this area, if you could partner up with some of these chemolithotrophs, that could be a very sweet situation. And so these tube worms do that. They have this relationship with these chemolithotrophs. And probably there's several Beggiatoa species of bacteria and Thiobacillus species of bacteria that they've been studying. And you can find these elsewhere. These bacteria, they're not just at the hydrothermal vent. You can go get Thiobacillus here in Marquette, no problem, if you know where to look. I haven't really hunted Beggiatoa before, but I sure have been able to capture Thiobacillus. Anyways, so you have these tube worms. That's largely what you're seeing in this image, but there's also these nice clams and mussels that are down by the hydrothermal vents, and they all have their own little suite of different chemolithotrophs that are capable of taking these inorganic minerals and CO2. And so it keeps the bacteria alive, but then the bacteria also gives some of that to the animal and they survive as well. So again, going back down into the deep ocean where we talked about weeks and weeks ago that this is the new idea, well, relatively new idea of where perhaps protocells evolved on Earth for the first time. And you've seen this already weeks ago, looking at the idea of the hot water, mineral-rich water coming up into these hydrothermal vent areas. And so there's many different vents, hydrothermal vents that they're studying around the world in the different oceans. Again, whenever they are getting down there with their submersibles and looking, they definitely see lots of life in that area. So here on the left, you're seeing a black smoker type of hydrothermal vent. So different minerals coursing through there with the heat creates this so-called black smoke, but they've actually named the hydrothermal vents. Some are called black smokers, and I can't remember what the other ones are called, but they have different names for all this. So they're teasing apart, the different environments. They're not all exactly the same. And then here is some of the bacteria that they isolated from the actual chimney area or the vent area. So the walls, it says the walls of the black smoker chimneys display a steep temperature gradient and contain several types of prokaryotes. So this one is, they call it a black smoker at the Snake Pit vent field of the Mid-Atlantic Ridge, 3,500 meters deep. That sounds big. Right. Yeah, I know, cold, right? Yeah, it's interesting. They have all these different names for the different vents. So yeah, definitely something I would like to experience at some point. Go down into those. They do take people down into the submarines. I'm sure they charge a pretty penny, but wouldn't that be cool to actually see something with your own eyes down there? You can get past the claustrophobia, I guess. Nice image again of the tube worms. And so we're going to zoom in a little bit and look at what's going on with them. So you see this pinkish region, pink-red region of the tube worm. So let's look at that a little bit and think about what's going on. So you have the bacteria hanging out in the tube worm and in specialized tissue. They call it the trophosome instead of a mycetome. They call it a trophosome. But basically, what's happening is the bacteria are taking the inorganic minerals. So here they're showing a sulfide, sulfur-containing compound and oxygen. And so this is what the bacteria are doing in the trophosome, all right? And so they're able to create sulfur-containing molecules that the animal, the worm, can take up. And the bacteria can also take CO2, and with stripping electrons off of those inorganic, they can make their own sugars. And so far, what they're noticing is that it's a very specific interaction. The tube worm, each tube worm species seems to only hang out with a very particular type of chemolithotroph. So far anyways, they're seeing only one species of bacteria per tube worm.
All the different species in the same family.
Of the chemolithotrophs? I don't know. That's a good question. I haven't kept up with some of the newer events they're studying to see if they've found that to be consistent or not. Is it gonna let me blow? I can't blow that up right now. What was the other thing? Oh, let me go on to the next slide here. So that red that you're seeing in those images, the red part of the tube worm, that's actually a modified hemoglobin. And so, we have hemoglobin as part of our red blood cells. This tube worm has a slightly different hemoglobin. What it does is, like our hemoglobin binds the oxygen and delivers it to our cells. The tube worm, it has hydrogen sulfide binding ability, oxygen, and CO2. And so it is specially designed to really help this interaction between the tube worm and the chemolithotrophs residing in its trophosome tissue. So it will use that hemoglobin to latch on to and bind to these molecules and then bring it to the bacteria, and then the bacteria use it. And so, for example, this would be a reaction that the bacteria, the chemolithotrophs, are doing, taking hydrogen sulfide gas and oxygen and creating sulfate. And you can see the free energy is huge, negative 798. You only need negative 32 to make ATP. And so you can see this is a big payoff, lots of free energy so that you can get ATP production. And so again, this is just an electron transport chain process that the chemolithotrophs are doing, just allowing for electrons to get passed from donor to acceptor. And in the end, you end up, in this case, with sulfate. And because you have all that free energy, you can get nice ATP production. And then the CO2, you can get that pushed into the Kelvin cycle, just like what we see with plants. All right, so here the chemolithotrophs also do the Kelvin cycle. Actually, they were the first to do the Kelvin cycle. The plants evolved that ability from the bacteria millions of years ago. And so it's quite a nice situation for these tube worms if, as long as they are getting full access to all the minerals coming through the ocean floor in that vent area, they can feed their chemolithotrophs that are hanging out in their tissue and get all their needs met. So again, a nice example of chemolithotrophs. Again, they, taking CO2 and fixing it, they are primary producers, even though they're not doing photosynthesis. Usually when you hear the word primary producer, you think about only organisms that do photosynthesis, but don't forget about the chemolithotrophs. So here again, just trying to impress on you, it's an electron transport chain process. You don't have to memorize this pathway. I'm just trying to show you, if you're a visual learner, that these chemolithotrophs are taking in different sulfur compounds and feeding it into some sort of electron transport chain system. And then that gives you a proton gradient, again, linking again the idea of an electron transport chain process to a proton gradient. And as the protons diffuse back across the membrane, that's what allows for ATP synthesis. All right, and then they're showing the CO2 getting taken and fed into the Kelvin cycle, which is the way that sugars can get made through the intake of CO2. Okay, so very interesting dynamic down in the hydrothermal vents, very expensive. If you want to do research with that, you have to have incredibly complicated and expensive equipment, including a submarine. So not cheap, but so much fun and interesting information coming from that research. Okay, let's get into this dynamic a little bit, looking at microbe-animal interactions and in particular with the ruminants. So that's just a name that we give to those types of animals that have a special GI tract that allows them to have like a chamber called the rumen. And when you peer inside the rumen, you'll find billions of different microbes doing very special activities. So these are ruminant animals, the typical cow that you think about, all the way to moose and caribou and giraffes, if you want to think about them. So as you have witnessed, I'm sure many of these animals, what they're doing is they eat grass and twigs and leaves, if you're a giraffe, I suppose, or antelope. And so that's cellulose. And so the plant is composed, the cell wall material of plants is composed of cellulose and hemicellulose and lignin. Well, those animals directly don't have the necessary enzyme to break that down, which is cellulase. So the animal, the cow, doesn't have that, but the microbes hanging out in the rumen do. So the rumen is essentially a sac-like chamber as part of the gastrointestinal tract, and it's anaerobic. And so only those organisms that are capable of, so you don't see aerobic or obligate aerobic organisms in here, but obligate anaerobes, facultative anaerobes, those will certainly be present. And so you have anaerobic respiration going on, you have fermentation pathways going on, and it is the way these ruminants eat their food, so they eat the food, let's say they're grazing on grass, the grass will get into their GI tract, the microbes will break down, and then they will regurgitate and kind of chew their cud type of idea. So as the cow or whatever is doing its eating, it will be eating a portion of the microbes as well. And so that's kind of a way to keep the microbe population, steady state, because you will get some of them being eaten along with the degraded plant material. And so this is research that has been going on for a long time, primarily having to do initially with the food industry, raising cattle and cows. So a lot of the veterinary medicine research has been looking into this. And so you can actually find cows that they've operated on where they have a portal. So this is usually capped. They have it uncapped right now, but usually this is covered with a cap. You take the cap off and then you can insert your arm right into the rumen of the animal. So they do this surgery to create a way to get at the rumen and then, wake up the cow and keep the cow happy and alive. But then they can keep doing time samples of going in and taking samples of the rumen by reaching in. So I don't know if you know Daniel Mitchell or not. He was a grad student in our department. He got his MSPBE degree here and also taught, I think Intro to Cell Molecular was the one he taught the most. But he took this class a couple of years ago, and when we started talking about this, he's like, yeah, I actually got to sample. I'm like, oh, do you have any pictures? So he was kind enough to share some pictures of him reaching in. Of course, you have to wear a very special, sterile, long glove to reach in there. This was down at Michigan State University. So here he's looking a little bit more happy. But that's what they do. They go in and they take these samples and then bring it to the lab and figure out, what's going on in the rumen. So there's been these cows can live many, many, many years under this situation, and they can do time course sampling with the same cow. And so what they're doing is examining the rumen. So you see here the gastrointestinal tract system of a ruminant. And so they're showing kind of the pathway of the food getting in to the rumen, and then it'll get back up into the mouth as like chewing their cud idea, and then back down. And so it all the way out to the small intestine and then the large intestine. So really kind of an interesting way of having their GI tract. Here's just another example. So with the rumen, depending on what publication you read, it can be 1 times 10 to the 10th to 1 times 10 to the 11th bacteria per gram. So in each single gram, you can have that many bacteria. That's phenomenally high populations. It's a nice home for the bacteria because it's stable. Temperatures, pH is pretty stable. And you can see it's slightly acidic typically. And here, this is just showing some fermentation ideas. So once you get to pyruvate, if there's no oxygen, which there isn't in the rumen, then they can either do anaerobic respiration or fermentation. So in this particular image, they're just showing different fermentation pathways that can happen in the rumen. So there's a mixture. There's definitely a robust mixture of microbes in there doing different types of physiology. But, you know, they're hugely important for the cow or whatever ruminant you're thinking of, because the animal has no way to break down that cellulose. And so that would be, you know, if you killed off the microbiome, you're going to have a very sick cow pretty quickly. Here is a little bit more detailed idea of what's going on. So here you see the ruminant bloodstream on the lower left here. So here they're trying to show how the microbes in the rumen are doing different physiological pathways, and then they're trying to show how certain these are actually organic acids, acetic acid, propionic acid, butyric acid. These are all organic acids that are getting absorbed by the cow, getting into the cow or the ruminant's bloodstream, and then it gets converted typically to proteins. VFAs, volatile fatty acids, these are also volatile, so organic acids, fatty acids. Eructation, that's the first time I heard that fancy word when I was studying about ruminants. That actually means burping. It's a fancy way to say burping. And so many people think that methane is actually going out the other end of the animal through gas coming out the rear end. It's actually the opposite. It comes out through their mouth through eructation. And so as they're chewing their cud, and they will release CO2 and methane. And so there's plenty of methane getting produced by the microbes so it's not the cow themselves. They, you know, they always give the statistic, you know, if you factor in all the millions of cows on the planet, and they're a huge part of methane gas release, and it's not really technically the cow, it's the microbes in the rumen that are doing that. So, but you can see here again that you do get significant amount of methane. The lower reaction here, 35 methane. All right, just so yes, they are contributors, definitely big contributors to methane production, but it's technically the microbes in the rumen. Okay, we got to stop there, and we'll keep going with some little more on this, and keep going with some other animals. Yeah, you know, they think if it's a vaginal birth, they are getting some microbes that way, just like they are seeing in the human state of the birthing process. And then just from, there are microbes in milk as well, so they're getting it that way too. And so it's a mixture of different ideas. Yeah. Okay. Here's my stop share. So we'll switch to lab. And it's not letting me get up into stopping the share. Interesting. Come on.
So I chose the kinds of pathogens